Well, in the words of Simon and Garfunkel, times they are changing. I think 10 years ago, if somebody had asked me about a cure for Parkinson's, I would have just stopped the conversation there. Even 2 years ago, I thought that was perhaps something that we shouldn't really be getting involved in talking about. But it is true to say that times they are changing, and we are now seeing an era, the first, certainly since I've been around Parkinson's disease, where we're really actually targeting things that might, if you like, treat the disease rather than just looking at symptoms and alleviating those. And I think first it's important to get our definitions right. So what do we mean by cure? I mean, cure is a four-letter word. And really when we talk about cure, we're asking a lot from Parkinson's because, as you know, by the time that somebody first comes to see a specialist like myself, complaining of symptoms of Parkinson's disease and gets the diagnosis, they've probably lost about half of the cells that produce dopamine, which of course leads to most of the physical symptoms and a lot of the other non-physical symptoms of Parkinson's disease. So the idea of curing a disease when already 50% of those cells are dead, gone, and unlikely to come back is a big ask. And I think that, we can talk about the idea of stem cells, and there are nice interviews with Roger Barker on my website, so you can go and have a look about where the field is with regard to that. And you'll have some talks, I think, also available online elsewhere. But really, I guess what we're focusing on is, well, at that point when someone's first diagnosed, is there a way that we'll be able to stop cells from dying? And I think, truthfully, if we were able to do that, and if I could offer that at the moment to the patients that I see, that would be as good as any cure goes out there, because as you know, all of our treatments at the moment target symptoms, and improving that quality of life is important. But all the time those cells are dying, the symptoms get harder to control with the tablets that we've got. People with disease often go on and will get problems with their memory and thinking, and delusions and hallucinations and falls and a whole range of other disturbances that if we could just stop the cells dying at a very early point, they may not go on to get. So I think we kind of have to accept the fact that if we could stop the disease or even just slow it so that it didn't end up advancing into those stages of the disease, that would be a big enough win for me right now, for sure. So I guess then the problem comes, well, you don't really understand Parkinson's disease. We don't understand what causes Parkinson's disease, and therefore it's hard to find a cure. We have some thoughts. We know that there are some genes that have been associated with developing the disease, and we'll talk a little bit more about those through this presentation. But the truth is that a mixture of what genes you've been born with and things you're exposed to in the environment still hasn't really provided an answer as to why those cells are dying. And therefore you might ask the question, well, how do we go about curing a disease when we don't know what's actually causing it? So then I guess we have to rely on the fact that over the last 201 years now since James Parkinson described the disease, what is it that we've observed? What knowledge do we have? And the truth is that the knowledge we have comes in lots of different areas. So, for example, if you look at something like the Sydney Brain Bank, which is an exceptional organisation that has done a lot of amazing research with the help of people mainly living in New South Wales, where they've donated their brain to science and research, and then researchers from all over the world can request that tissue. Well, that itself has provided some clues. So we have some idea as to what the brain looks like in Parkinson's disease. So we know that inside the cells that are dying, there seem to be changes. And one of the commonest ones people talk about is the appearance of Lewy bodies. And Lewy bodies, essentially, looking down the microscope into those cells that are dying, are these balls of tangled protein called alpha-synuclein. Now, I always think about this a bit like clothes. So if you fold your clothes nicely, and the clothes here are protein, and you put them into a drawer, the drawer will close nicely, and that's a bit like our cell working well when the cell's proteins have got the right configuration. But if those proteins become tangled, like tossing your clothes into a drawer, the drawer doesn't close properly and the cells may die. Now, that's a very rudimentary understanding of what might be going on, why you find these proteins inside the dying cells, and we don't quite know that's the case. These cells maybe you have the protein in there because the protein is trying to help, but we have observed these things. And we do see this pattern of cell death associated with these animal proteins. So some clues there. And then from the world of, if you like, more basic science research, or animal research and models of Parkinson's disease, and it is important to point out that animal models are just that. The fact of the matter is that animals don't get Parkinson's disease. The only animal on the planet that gets Parkinson's is humans. But for our purposes of trying to understand the processes that are going on in Parkinson's, those animal models can be very helpful. So from those, we can actually see things that might be causing or leading to cell death. And so we understand things a bit more now, so we can see things like processes like inflammation going on in and around the brain cells. We can see things like the energy consumption of those brain cells being important through the powerhouses of the cells, the mitochondria and the pathways that support those mitochondria that give the cell the energy that it needs. And also understand a little bit about what the cell is doing. You know, we talk about producing proteins. Proteins in this case aren't like, you know, cheese and meat that we eat in our diet. This is us making these molecules out of the building blocks that we do take from our diet, but effectively configuring them in special ways. Maybe they become an enzyme, and that enzyme does something. So just like in the gut, enzymes digest food and break it down so you can absorb the molecules. Well, here at the cellular level, maybe those enzymes work in a way to, if you like, clear garbage, maybe those tangled proteins, and stop them from tangling or clear them out of the cell, emptying the garbage. So we are seeing from the basic scientists some real advances in understanding why those cells might be dying. And we touched earlier on the idea of genetics. And so genetics, we know that really the people who have a gene that causes Parkinson's make up a very small number of the cases that we see. Maybe less than 2% of all of our patients happen to have a gene that you can say, yep, that's what caused your Parkinson's. But those genes do give us insights, because the genes will code proteins, and the proteins, if you like, may become enzymes that give us a clue as to why those particular genetic abnormalities are causing Parkinson's. So perhaps those genetic abnormalities lead to the deficiency of an enzyme that might clear garbage from the cell. And interestingly, probably the commonest genetic abnormality we see in cases of Parkinson's disease, which don't appear to run in families, is this gene called glucocerebrosidase. And this shortens down to GBA, and there are other great people around giving lectures on this. Nick Zanko, I think, will cover a lot of this work. Hopefully you'll find some of his stuff online too. So the idea here is this enzyme seems to be deficient. Now, interestingly, we do know of a disease where if you don't have any of this enzyme, you're in real trouble. And this is a condition called Gaucher's disease, or Gaucher's disease, where effectively children usually develop problems where they can't clear abnormal proteins from cells, and they get real problems down the track and present in various ways. And that's if you've lost both of those copies of the gene. But in Parkinson's patients, about 5% of them will have lost one of those. So they are what we call heterozygotes. So they haven't lost both of the genes, but their ability to produce that enzyme that clears garbage from the cells is reduced. And what we are seeing is that that may account for a whole bunch of patients who are getting the disease. And of course, combined with other factors, whether it's inflammation or whether it's the lack of energy, there may be cascades. In fact, there may be more than one process leading to Parkinson's disease itself. In terms of understanding the disease and getting clues as to targets to treat it, as well as those things from the basic science, we've also got things from what we call clinical epidemiology. That is to say, well, look, what's out there? What can we observe amongst patients who've got the disease? Are there any clues there that might lead us to an insight into what to target with, say, a drug trial? The commonest or the most famous of these relationships, of course, is smoking. And smoking, believe it or not, reduces the risk of getting Parkinson's disease. Now, before you all go rushing off and starting to smoke, please remember that the increased risk of getting lung cancer and chest diseases is very, very high. The reduction of getting Parkinson's is said to halve the risk, which means you go from a 1 in 1,000 to a 1 in 2,000 chance of getting Parkinson's disease. But it does raise the question, what is it about cigarette smoke that might protect you? And in actual fact, people have started to look at things like nicotine, not so successfully, but people have looked with trials, as it were, to see whether nicotine might slow your chances or progression of Parkinson's disease. And those studies have, at this point, not shown any benefits, but they show you again where people are targeting their thinking. And possibly the most recent story in this category, if you like, is an observation that was made about drugs like beta blockers, so the drugs that people take to control their blood pressure. They seem to be associated with an increased risk of getting Parkinson's. Now, if you're on a beta blocker and you already have Parkinson's, well, in some ways the ship has already sailed, so please don't go stopping your blood pressure tablets. Interestingly, on the flip side, the opposite kind of drug action, a thing called a beta agonist, so beta blockers lower your blood pressure, but beta agonists are the things that are inside those blue inhalers for asthma. People who take those seem to have a reduced risk of getting Parkinson's. So here we're talking about a clinical observation where we're saying, well, what is it about this pathway around what we call the noradrenergic system? And interestingly, some researchers have had a look to see what these drugs might do to the biology of the cell, and have found things like an upregulation or a downregulation of a thing called the synuclein gene. So that synuclein protein perhaps being, if you like, influenced by medications. So we can see how we might get insights into strategies for treating a disease. And I can't cover all of them today, but I want to give you some sense of what is out there and where people are looking. So in terms of coming up with, if you like, a drug treatment, and so leaving aside things like exercise therapy or other dietary changes, I think we'll stick to pharmacological approaches. There are basically two main areas. So if you like, there are the idea of coming up with new molecules, something that's novel, has to be invented, and then the idea of a repurposed drug. So a drug that already exists being used for a different indication. And we're going to talk about both of those next. So the best example I can think of in the field of new molecules relates to some of our understanding, again from the biology of the disease of Parkinson's. And the idea that Parkinson's might in some ways be a bit like Creutzfeldt-Jakob disease or mad cow disease. And this idea is called a prion-like hypothesis. So the abnormal changes in the brain of people with those diseases is, if you like, tangled proteins, the theme of the day, as it were, and being triggered by something that might not have been abnormal in the first place. And that's something you might have to get into your system. Either you ingest it, eat it, or inhale it, you breathe it. And we know from the epidemiology that people with Parkinson's often will have problems with, say, their gut, with constipation, or their sense of smell, long before they develop the physical symptoms of Parkinson's. And the idea is maybe something gets into your system, then crawls along the nervous system all the way up to your brain, and in actual fact there are changes in the olfactory bulb where the nose, as it were, receives the input for smell, and also in the area of the brain where the nerve that goes off to supply your gut enters the brain or leaves the brain. So some, if you like, circumstantial evidence. In addition to that, in the early noughties, people were looking at trying to treat the symptoms of Parkinson's using stem cells. And in this case, stem cells reflect actually fetal cells. So cells that were taken from aborted fetuses, so some ethical issues already there. The idea of taking the area of the brain that was going on to produce dopamine, collecting those cells, and then putting it into a patient with advanced Parkinson's disease in the hope that those cells would produce dopamine in a more, if you like, biological, physiological sense. Now, those studies were actually stopped early for safety reasons. The patients involved in the trial actually developed what we call runaway dyskinesias. That is to say, they started developing involuntary movements even when you stop their dopamine tablets. And those patients all needed to go on and have deep brain stimulation surgery to try and control their symptoms. But the researchers pursued those patients, and now some of them have actually come to post-mortem. And when you study the brain cells and look at those cells that came from the fetuses, which clearly never had Parkinson's, inside them there is evidence for Lewy bodies, so tangled alpha-synuclein protein, which one would have to argue must have come from the host. So if you like, infected, or the term used, transfected, from one cell, i.e. the person with Parkinson's, into those cells that came from the fetus. Now there are some caveats. We know that in our brain bank, we look at the part of the brain that had the changes. In Parkinson's disease patients, you never see these tangles of Lewy bodies in that part of the brain. So you have to accept something different has happened here. But it does spawn the idea that there is a possibility that, if you like, the alpha-synuclein protein that we've been talking about may be a bit like a prion and spread from cell to cell to cell to cell, spreading through the brain, giving increasing symptoms over time. So probably the most exciting area that's being pursued in the new molecule space, and there are other new molecules I'll grant you, but is the idea of targeting this alpha-synuclein prion type model. And there are a couple of ways of doing that. So one of them is what we might call a vaccination program, so active immunization. So if we could expose people who don't have Parkinson's to an agent that might protect them from synuclein protein, that is to say, give them a little bit of synuclein protein so that they don't react to it when they get to see it later in their life, then that might be a way of stopping Parkinson's. Now, that approach is very big, because what you'd have to do is to possibly immunize everybody, a bit like whooping cough, and your return on that model is pretty low. So the alternative approach is what we might call passive immunization. And people will have come across this for other diseases. So diseases like rheumatoid arthritis or Crohn's disease currently have treatments where you produce antibodies, monoclonal antibodies, synthetically, and target them against the thing that you want to block. And in this case, you'd be trying to block the synuclein, either trying to stop it from aggregating or perhaps stopping it from transfecting one cell to the next. So we have this approach, and there are trials ongoing in America, which are safety trials at the moment, which have got industry backing, and these trials will cost probably billions of dollars. And of course, it's a very expensive strategy. It's a very high-risk strategy. It might not work. In fact, taking the alpha-synuclein protein out or blocking it might make things worse if the alpha-synuclein was trying to be, if you like, a bodyguard for the cell. And the other thing is it's going to be slow, because we know that these treatments are going to have to go through a lot of regulatory things to stop from being dangerous and getting to the market. So then we look at the alternative approach, if you like, the idea of repurposing drugs that we already have. So here what we're talking about is the idea of saying, well, if we know something about the cell biology in Parkinson's disease, and let's say that we think that inflammation is important, then we've got any number of anti-inflammatory drugs that are around, and therefore you might say, well, why don't we target that pathway? Find an anti-inflammatory drug that's available, we know it's safe in humans, and do the trial. And this can be thought across a whole bunch of other things. So if you like, increasing the energy supplies to the cells through the mitochondrial pathway, through improving like the energy systems, clearing garbage out of the cells. And indeed, these approaches are happening. So the most recent and probably most famous of these was Tom Fulton's trial in London, published in The Lancet just last year, using a small number of patients, just 30 on placebo and 30 on an active drug, which in this case was an injection for diabetes treatment called exenatide. So not insulin, but something that operates through a different mechanism. And in that small study, what Tom and his team were able to show were differences between patients who got the drug versus those that got, if you like, a water injection. Now, personally, I think there are some things that we need to keep in check before we get too excited. It's a very small study, and Tom himself admits this. And the other thing that I think was interesting about the trial for me was if I saw a disease-slowing drug, what I'd expect is for everybody to get worse, but those people on the effective drug to get worse more slowly. So effectively everyone's on the decline, but the steepness of that curve is different if you've had a treatment that's effective. And what the exenatide trial showed was a little bit different. In actual fact, their study showed that the patients on the drug seemed to actually improve in their Parkinson's symptoms while they were taking the drug, and those symptoms then got a little bit worse when they stopped taking the drug at the washout period. Now, I think it's very encouraging, I think it's an excellent thing, and it's definitely triggering a larger trial that's going to be international, and people are going to look at this drug very hard. But we do have that little caveat as to whether it really is slowing the disease. But what it does do is it gives us a roadmap something that can allow us to say, okay, well, if we were going to do a trial to look at repurposing these, if you like, drugs that exist for Parkinson's, what trial would we do? And the truth is, these trials are actually already here in Australia. So there are a few centres around Australia that have been asked to take on trials for industry, so drug companies coming along. And some of these will be using novel molecules, and some of them will be repurposing medications that exist. And if people are interested in looking at what trials are available here in Australia, there is a website. It's www.ANZ for Australia and New Zealand, Clinical Trials Registry, so ctr.org.au, so www.anzctr.org.au. And you can actually type into that website Parkinson's, and it'll show you the trials that are coming up in the Parkinson's field here in Australia. So I think most of these trials that have come from industry are interesting, and what they're doing is generally targeting people who are newly diagnosed, who aren't on lots of medications, if you like, at the start of their journey. So I think that this is going to be very exciting. But what I want to go on to talk about next is, if you like, one of the main reasons that we're interested in this whole virtual environment to spread news about work that's going on, and that's the proposed linked clinical trials initiative program here in Australia. So in Australia, this is being supported by the Shake It Up Foundation, and the idea is to get people from all over the world. So Shake It Up have partnered with people like the Cure Parkinson's Trust in the UK, Michael J. Fox Foundation, the Garvan Institute, Grand Rapids Institute in America. And what happened a few years ago is that a bunch of basic scientists, researchers, and clinicians sat down and said, look, we understand as much as we do about the disease, what drugs exist that might target some of these mechanisms we've been talking about that we could safely use in a clinical trial? And so Shake It Up have approached me to lead quite an ambitious program here in Australia. It's ambitious because what I'd like to do is to not do a standard trial where you take one drug and compare it to a sugar pill, because I think that's very good, but what I think we're in the business of is trying to screen as many of these drugs as we can as possible. And the proposal at the moment is sitting with a review committee based in the States, who have got members on there who are involved in clinical trials and drug regulation and toxicology, really having a look to see what we're proposing is going to be feasible, and so we're doing our due diligence. And the idea is to actually have not one, but 4 active drugs and 1 placebo arm. So effectively, if you enter the trial, you might get a 1 in 5 chance of being on the sugar pill, but a 4 in 5 chance of being on one of the active drugs that we hope might slow the disease. Now, we don't expect such a trial to answer the question, is this a drug that's going to cure the disease? The answer from this trial is going to be twofold. Really, what we're aiming to do is to show Australia and the rest of the world that we can do trials. We have a large population of patients who are dead keen to get involved and volunteer in this work. We also have a fantastic, skilled workforce of clinicians working here in Australia who have not had the opportunity to run these sorts of trials. So I'm hoping that this work will serve as a roadmap that will be spread across Australia, and if this is the last of these trials that we do, then I suspect I will have failed. I think our aim here is to trigger the next trial, and if we see one drug that's successful, go and ramp it up and do that bigger multi-centre trial that's going to be important to proving its efficacy. The other aspect of the trial, which I'm very keen on, is the fact that we're partnered with some of my basic science colleagues here in Australia who are much smarter than I am. And these are people who are going to set up 2 things that are important for us. And they're both really blood tests. One of those aspects of the blood test is to see whether there is what we call target engagement. If we pick a drug that we think might change inflammation, on a blood test, can we show that that's what happened when people started taking this drug? So target engagement we should be able to get from our basic science colleagues. The other really exciting aspect is that working in collaboration with these people, and Michael J. Fox funded studies, again supported by Shake It Up, we've actually already started to develop what we call biological markers of the disease. So if you like, disease markers. Markers that we can see are different between patients and people who don't have the disease. This might be measuring the activity of an enzyme that might clear, if you like, protein or garbage from the cells, or indeed measure things like the buildup of synuclein so that we can have an idea of monitoring that protein. So in the trial we're going to be able to not only look at whether the target engagement is accomplished, that is to say, did we change inflammation or energy pathways, and also did we actually do anything to the biology of the disease? Because at the moment we rely on just having clinical assessments where patients are having to do physical things, move their hands, walk, etc. as our outcome measure. So what I'm very confident that we'll be able to do in this first trial is to get a roadmap so that we can do more trials, and also get a biological panel that the rest of the world will start to use, so that we will get some outcome from this trial, even if none of these drugs are shown to be effective for treating Parkinson's disease. So I think that's very bold, and I think that's very exciting, and I think we can get involved. Our timelines, I'm hoping that we'll have this running before the end of the year, and people can keep track of this through my website and also through Shake It Up Foundation. Our initial studies will target people with more advanced disease, who've had the disease for a few years and are on more medications. And the idea is if we can see it working there, we'll take it to people who have earlier disease, and also people who may yet develop the disease. Many of you will know my research looking at a thing called dream enactment behavior, where if you have that, your risk of developing Parkinson's is over 100 times greater than the baseline. So in actual fact, what we're hoping to do, if you like, is have this overarching theme where we can have a look at trying to fight back against this terrible disease and offer hope to everyone with Parkinson's who is affected, living here in Australia and around the world. Thank you so much for your attention.